All right, anatomy and physiology class. Welcome to TBL day number 2. This is the male reproductive system. So we'll start off the TBL activities watching this fertilization video that you see there in the background. And it really is this incredible, beautiful story, this journey that is the beginning of all of our lives. And we'll start out with that. But for these notes, we're going to dive right into just some kind of review on the differences between mitosis and meiosis. And so mitosis is what most cells, like somatic cells, body cells, go through, where there's a single division of cells. DNA is replicated before that division happens. So then when we get the two new daughter cells, each of those daughter cells have the full number of 46 chromosomes, or 23 pairs. Okay, so we got 23 pairs, or 46 total in each of those. Okay, but then we go over to meiosis. With meiosis, there is DNA replication that happens beforehand. So that means we start out with double the amount of DNA and chromosomes. But then we have a first division in meiosis 1, okay, and then that gives us two daughter cells that start out with 46 total chromosomes. But then those split, and there's no more DNA replication that happens there, and so we end up with 4 cells with 23 total chromosomes each. Okay, and those cells are known as haploids. All right, so that's a little bit of the basics there. Let's go through just some differences here between mitosis and meiosis. So I just kind of said this on the last slide. Somatic cells for mitosis, germ cells, which are eggs and sperm, also known as gametes for meiosis. Daughter cell resemblance to the parent cell, genetically identical for mitosis, not identical for meiosis. Okay, daughter cells resemblance to each other. Oops, sorry. Genetically identical, but then not identical for meiosis. Number of chromosomes, daughter versus parent, same, but then again, half the number of chromosomes in after meiosis 2. Okay, the purpose of mitosis is to grow, to replace dead cells, aging cells. Meiosis, the purpose is reproduction. Okay, so germlines are the precursors to egg and sperm cell. We'll talk about the germline in the testes today, the spermatozoa. Males have a mitotically dividing germline. That means the germline cells go through this process of mitosis. They're constantly replacing themselves, okay? And so then we're constantly getting new sperm, okay, in males. In females, there's mitotic division of those germline cells that are going to make the eggs, but only during fetal life. Once female is born, there's no more mitosis happening in that germline, and so really all the eggs that a female starts with is the most that she'll ever have. Okay, so again, in meiosis, the daughter cells that are made have only 23. Oops, sorry about that. Have only 23 total chromosomes. Daughter cells made in mitosis have 23 pairs, but really 46 total chromosomes. The 46 chromosome number is restored in those gametes, in those sperm and egg cells, really, but it's not in both. It's when they come together, right? When the egg gets fertilized by the sperm, 23 from the egg, 23 from the sperm come together, and now we're back to our 46. Okay, so a little bit about male development. As I just mentioned, during fetal life, there's a mitotically dividing stem cell population. Okay, so we're increasing the size of the stem cells. Sperm stem cells are called spermatogonia. Okay, and I think I said spermatozoa on the previous slide. That's not the correct term. Spermatogonia is the correct term. Okay, so then after birth, mitosis stops all the way up until puberty, and then mitosis begins again. Okay, so there's a period of time between birth and puberty where there's no mitosis happening in the spermatogonia, but after puberty begins, mitosis starts again, and that continues on through the male's life. During development, the testes descend into the scrotal sac. This typically happens before birth, sometimes after birth. Sometimes, as you can see here, so this is that inguinal canal where the testes descend through. Sometimes that canal doesn't close up all the way. It's supposed to close up enough to where it's mostly sealed. But what happens sometimes when that doesn't close up all the way is you get something called inguinal hernia. Okay, and what the deal with that is, so the spermatocord passes through that inguinal canal, so obviously it doesn't close all the way, but it needs to close enough to where when a male pushes down and bears down or lifts a heavy weight, the abdominal cavity, the intestines in the abdominal cavity can actually push against that part of the pushes against the abdominal wall. And if there's a big enough hole there, those intestines can actually squeeze down into the scrotum, and that's what can happen with this herniated intestines here. Okay, so this can be surgically corrected if they don't close all the way. And that's what's known as an inguinal hernia. Okay, so then starting back up at puberty and adulthood, stem cell generation regenerates again via mitosis, and stem cells differentiate into sperm, and they go through meiosis and multiply that way. Okay, so let's dive into a little bit of the male anatomy. So the scrotum is really just an outpocketing of the abdominal lining. And so really the testes need to exist outside of the abdomen. Okay, and the purpose for that, one of the purposes, is to keep the temperature a little bit lower than body temperature. Okay, and there's problems if testes are held too close to the body with very tight underwear at times, or sometimes with hot tubs and things like that, where testes undergo much hotter conditions than they're designed for. This can lead to some infertility problems. Okay, there's actually two specialized muscles to help the testes maintain the correct temperature. And those are called the cremaster muscle and the dartos muscle. The cremaster muscle actually lifts the testes up themselves, and then the dartos muscle is more connected to the externally, and that pulls the testes closer to the body by scrunching the skin in the scrotum towards the body. Okay, the little bit more of the anatomy in the testes. So there's a membranous structure, protective layer called the tunica albuginea. This is the white protective layer inside the testes. This surrounds what's known as a seminiferous tubule, which is a set of convoluted tubes that is really the beginning of sperm development happens here. Okay, this is where those spermatogonia cells would be found. And then after that, we've got the epididymis, and this is a series of ducts in the posterior portion of the testes. This is where sperm mature and they become motile. Right? We could say they learn to swim. All right, and then from there we get up into the vas deferens, or the ductus deferens. This is the connection between the spermatocord and the epididymis, and descends through the inguinal canal into the abdominal cavity. And this is surrounded by blood vessels, nerves, and smooth muscle. During the ejaculation process, smooth muscle contraction squeezes semen out of the vas deferens and the spermatocord towards the urethra. Okay, so a few of those structures here in the testes. We can see kind of a cross section of the testes. So that top structure in A, so actually what I want you to do is go to figure 26.4 and fill in these structures here to make sure you're familiar with those. Okay, more about the male anatomy, the ducts and glands throughout. The ejaculatory duct is where the vas deferens and the duct of the seminal vesicle meet. So this is right before it turns into urethra. Okay, this wall is lined with smooth muscle, which helps with ejaculation. Then the urethra is the duct for semen and urine. So this is the first kind of shared tube system, right? So this is between the urinary system and the reproductive system. We'll see in females later, there's really no shared tubular system, but in males there is. And then we see the seminal vesicle, which is posterior to the bladder. This is where the vast majority of semen, the volume of semen, comes from. 70% of it comes from the seminal vesicle. It secretes a very alkaline fluid, also with fructose. Okay, fructose is a sugar, so that's going to be food for the sperm to go to its mitochondria and make ATP, so it can swim very rapidly. And then the alkaline fluid is there to neutralize the low pH found in the vagina, as well as in the urethra of the penis. Prostate gland is going to be below the bladder. It surrounds the urethra. It seems to grow continuously in males. and it blocks, if it grows too much, it can block the urethra. It's also very prone to cancer. Okay, it secretes a thin, slightly acidic fluid that we don't really understand the purpose of. And then lastly, the bulbourethral glands. We'll see similar glands like this in the female anatomy as well. Very small, pea-sized, inferior to the prostate. They secrete more of a thick, clear mucus. And this happens prior to ejaculation, so not part of the semen, but the juices that come out prior to ejaculation. So this is there for lubrication, again, neutralization of the pH of the urethra prior to ejaculation. Okay, so again, you guys can go ahead and go to this figure, label those structures that you need to label there, make sure you're familiar with those. All right, and then lastly, anatomically, the anatomy of the penis. There's three regions: the root, the body, and the glands. And the glands is covered by what's known as a prepuce, which is the foreskin. Okay, this is the piece of skin that's removed in about 60% of American boys, but only 15% of boys worldwide. Okay, so this is a highly American thing for this to be more normal, for to have a circumcised penis, but across the world it's very, very rare actually to undergo circumcision. So much so that most insurance companies don't cover circumcision anymore. It's an elective thing that you actually have to pay for, and your insurance won't cover it. And there really is no scientific evidence to show that it's safer or more hygienic or anything like that. All right, when it comes to vascular anatomy, it's sort of interesting. There's deep central arteries that are surrounded by these big blood sinuses or cavities. So during the erection process, the parasympathetic nerves are stimulated. Those lead to nitric oxide release. Nitric oxide is a very strong vasodilator, so that causes those central arteries to dilate. And what that does is those arteries then fill up those sinuses that are surrounding the arteries, which then that smashes down their ability to drain. And so the blood gets stuck in there, right? And that leads to engorgement of the penis. Okay, erectile dysfunction, or ED, is extremely common in westernized countries, and it's 50% of men, so half of men over the age of 40 struggle with erectile dysfunction. The most typical cause is damage to the artery walls that just happens with aging, and that's directly connected to nitric oxide release. Okay, something that's seen more in younger males more recently, which is a very sad story about the state of our society, is something called pornography-induced erectile dysfunction. This is the idea that more of a neurological disorder, to where a young man is used to seeing such extreme images in pornography that when it comes to an actual sexual encounter, that's not enough stimulation to lead to erection, and that's become somewhat of an epidemic in teens and males in their 20s. And so the treatment for typical erectile dysfunction, not pornography induced, but typical, would be Cialis or Viagra, which you've all heard a lot about. And these are directly connected to increasing or enhancing the effects of nitric oxide. Okay, so I'll let you go ahead and fill this in based on the textbook image as well. Figure 26.5 again, filling in those labels there. Okay, so when it comes to the ejaculation process, semen is the name of the fluid that's released, and it's typically about 2 to 5 mils of fluid. Okay, only about 10% of that is sperm, but in those 2 to 5 mils, there's somewhere around 20 to 150 million sperm per milliliter of semen. Okay, so lots and lots of sperm per mil of semen. Okay. Outside of the sperm, there's also acid buffers to neutralize the pH in the urethra and the vagina. Those are typically acidic. Okay. There's also fructose. Again, this helps to provide fuel for the sperm. There's also prostaglandins. Prostaglandins stimulate uterine contraction, which helps to kind of move sperm along, as well as to start labor. And so one of the common things that OB/GYN will tell a pregnant woman who is tired of being pregnant and ready to get that baby out is, as they will actually recommend sexual intercourse with their spouse in order to potentially kick off uterine contractions and start labor. Um, there's also clotting factors to make the semen stick to the walls of the vagina so it stays inside. And then finally, that ejaculation process is a sympathetic process. And as we talked about with the autonomic nervous system, erection is parasympathetic, whereas ejaculation is sympathetic. So there's kind of this blending of the two systems that we don't normally see very often. Finally, the term vasectomy is the surgical cutting and tying of that vas deferens tube. Okay, this is considered 100% effective birth control, also known as sterilization. Okay, so it's common for males to have this done after the couple is done having children. Problem is, if they decide they want to have some more children later, it's only 50% successful to be able to reverse it. Okay, so a little bit about the process of sperm development, which is known as spermatogenesis. There's two important cell types that you'll need to know about. So the Sertoli cells and the Leydig cells. Okay, Sertoli cells are also known as sustentacular cells. Okay, and so we can see those here, right? And really, those are large cells that kind of take up this whole region between the sperm cells. Okay, and then the interstitial cells, also known as Leydig cells, are up here. They're kind of behind the membrane in this other layer that don't necessarily interact directly with the sperm cells, but they're kind of behind. Okay, so the Sertoli cells are kind of known as the nurse cells. Their job is to take care of the sperm that is surrounding those sperm. So they secrete all kinds of different substances that aid in the growth and development of sperm. They also form what's called the blood-testes barrier. And the reason for that is because those sperm cells, remember their DNA is different than a typical DNA of a cell in the male's body. And so if the immune system was able to interact with those sperm cells, the immune system would actually destroy those sperm cells. So we need to have a barrier between the blood and the sperm, and the Sertoli cells help do that. And then the Leydig cells, or the interstitial cells, again up here, those ones are kind of around the seminiferous tubules, back in the connective tissue, and their big job is to secrete testosterone. And they're the primary source of testosterone in males. Okay, so let's walk through the hormones a little bit here. We've got LH and FSH, so luteinizing hormone and follicle stimulating hormone. Luteinizing hormone in males goes right to those Leydig cells that we were just talking about, stimulates them to release testosterone. So that's the primary thing that happens with LH and the Leydig cells. So you can remember L goes with L, Leydig, LH. Okay, and that stimulates the release of testosterone. All right, FSH, or follicle stimulating hormone, again, these are named for what they do in females, not for what they do in males. There's no follicles in males, but what they do is they stimulate those Sertoli cells to release those chemicals to aid in sperm development and production, as well as they stimulate Sertoli cells to release a hormone called inhibin, which is going to provide some negative feedback up above in the chain of hormones, and we'll see that here in a second. Testosterone is very important for sperm development, as well as producing secondary male sex characteristics like facial and pubic hair, deep voice, and increasing muscle mass. And lastly, inhibin, again, is providing negative feedback to inhibit the release of GnRH, FSH, as well as LH. Okay, and that kind of just helps us to control the rate of sperm production. It doesn't cause an overproduction of sperm. Okay, so the male sex hormone control loop here is an important one to walk through. So again, we're always going to start with a hypothalamic hormone, right? And so that hypothalamic hormone is going to be starting up top, okay? And so that is going to be our GnRH, gonadotropin releasing hormone, okay? That's going to come from the hypothalamus down to the anterior pituitary. The anterior pituitary is going to release both LH and FSH. Okay, again, LH is going to stimulate those Leydig cells, but number two is actually the other one, which is FSH. FSH is going to go down and stimulate those Sertoli cells. Okay, stimulate sperm production. Okay, so we can see that in the Sertoli cells, and then number four is going to be our LH. Remember, L goes with the Leydig cells. That's going to stimulate Leydig cells to release testosterone. Okay, and then we can see number 5 there is the testosterone. That's going to go cause all of these effects, right? Somatic, psychological effects at other body sites, secondary sex characteristics. And then we see these are the negative feedback loops here, right? Um, Number 6 is the testosterone, providing some negative feedback, okay, to both the hypothalamus and the pituitary, okay, and then we can see inhibin also providing negative feedback directly to the pituitary gland, stopping it from releasing too much LH and FSH. Okay, so when it comes to anabolic steroids, this would mean somebody giving themselves extra testosterone above and beyond what their testes produce on their own. That's one way to do it. Also, more commonly lately, with the baseball players that have been caught doing this in the past decade, instead of just giving straight testosterone, they give precursors to testosterone, known as androstenedione. And we talked a little bit about that already in the endocrine system section. And so, but one of the problems with this is the hypothalamus senses high testosterone. So does the anterior pituitary, because we were just talking about how testosterone is one of the primary negative feedback mechanisms that we have here, right? So if we have high testosterone, that provides negative feedback, so it stops the release of GnRH, stops the release of LH and FSH. Now we might think, well, that's fine because we got enough testosterone, we don't need to do that. But these two hormones did a lot more than just cause testosterone release, right? That was one effect of LH, but FSH was very important for sperm development, right? And very important for maintaining those Sertoli cells to maintain the blood-testis barrier. And so that can lead to infertility. One of the reasons that steroid use can lead to infertility. Okay, so some side effects of anabolic steroid use: infertility, and we just kind of walked through that. And then secondly, cardiovascular disease. This can lead to high cholesterol, as well as an unhealthy thickening of the heart muscle because testosterone causes muscle growth. The liver is responsible for metabolizing those steroids, and so if we've got too many of them, that can cause liver damage. Testosterone can lead to emotional changes, so rage, instability, depression. Can also end up with gynecomastia, which means breast development in males, because what happens is there's just so much excess testosterone around that can be converted into estrogen, and then you can start seeing female sex characteristics. And then lastly, ulcerated acne. Those sebaceous oil glands we talked about last semester, those are directly stimulated by testosterone. So if they just start overproducing with too much testosterone, can lead to those infections, which leads to acne and can be serious acne like you see here. All right, guys. Well, that's it. Study this hard, and we'll see you when you're ready for the IRAT.